Okay.
Hello, everyone. Good morning. Welcome back to our e-seminar series on Translational Biomedical Engineering. Today, we have a great pleasure to host Professor Nasim Annabi from UCLA with us. Before we start, I would like to go over a few housekeeping notes. So, first of all, I would like to apologize for the typo that we had in the announcement for Professor Annabi yesterday. Thank you for those of you who connected us to correct the date. We apologize. Sometimes these things happen, especially after a year of running these e-seminars. But I would like to apologize again for this typo. As usual, if you have any questions during the talk, please write your question in the ask a question box so we can track your questions and then ask them from the speaker. Also, we have a poll. Please participate in the poll and let us know what you think about these e-seminar series. Of course, you can always email me and Human and share your thoughts about these e-seminar series with us. So I'm very pleased to announce that we have a list of fantastic speakers for the summer. We will start with Professor Annabi, but we have many excellent speakers who agreed to give a talk at this e-seminar series. Please follow us on Twitter and then also on the LinkedIn to get the most up-to-date information about these seminars and then the next speakers. Specifically next week, we have Caroline Baudot from Polytechnique Montreal, who will give a talk on translating innovations in biophotonics from lab to market and back to the lab. We're looking forward to Caroline's talk next week. Also, as usual, I would like to thank our sponsor, Montreal TransMedTech Institute, who has been with us since almost the beginning of these seminar series. Thank you, Montreal TransMedTech, for supporting this initiative. So with that, I would like to invite a friend, a role model, a mentor, a former mentor of myself, Professor Nasim Annabi, to give a talk. Nasim is an assistant professor in the Department of Chemical and Biomolecular Engineering at the University of California, Los Angeles, UCLA. She received a PhD in chemical engineering from the University of Sydney in Australia. From 2011 to 2014, she was a postdoctoral fellow at Harvard Medical School and the Wyss Institute for Biologically Inspired Engineering. That's where I got to know her and then learned a lot from her. She was my first mentor back then. Before joining UCLA in 2018, she was an assistant professor in the Department of Chemical Engineering at Northeastern University. Dr. Annabi's group has expertise in the design and engineering of advanced biomaterials for applications in regenerative medicine. In addition, the research team has devised innovative strategies for the development of advanced bioadhesive and surgical sealants with high clinical translation for surgical applications. Dr. Annabi has published over 135 articles in prestigious journals. She has been cited over almost 13,000 times and has an AIChE index of more than 55. Her innovations have resulted in 15 patents and generated significant commercial interest. Dr. Annabi has been recognized with several national and international awards, including the 2021 Young Investigator Award from the Society for Biomaterials, a very prestigious award, the 2020 Nanoscale Science and Engineering Forum Young Investigator Award of American Institute of Chemical Engineers, the Australian Prestigious Endeavour Award, and the National Health and Medical Research Council Early Career Award. Her team has received major grants from the National Institute of Health and the Department of Defense and the American Heart Association. So with that, I would like to invite Nasim to share her screen. And Nasim, the virtual floor is yours.
Great. Thank you so much, Mohsen, for the nice introduction. Let me first figure out if I can share it again. Do you see my screen?
Yes, we do
Okay,
see,
great.
and we see your presentation. Yeah.
Perfect. Thank you very much. So before I start, I would like to thank Mohsen and Hooman for organizing these great e-seminars. I know that it's great for many of us during pandemic to hear from different people. And I love the topic of these e-seminars because I'm highly interested in translational research. This has been the focus of my lab, and I believe that these series are very, very useful for everyone. So today I'm going to talk about a multifunctional bioadhesive hydrogel for medical application. Before I go through the topics that I want to discuss today, I would like to give you an overview about my research. My lab focuses on engineering 3D hydrogel-based biomaterials with tunable physical and biological properties. Once we form this hydrogel, we combine them with different nanomaterials and nano-delivery platforms, and also different microscale technologies. And at the end, these microfabricated hydrogel are used for tissue engineering application. We are very interested in soft tissue engineering application of this hydrogel. And if we can improve the adhesive properties of this hydrogel to the wet surfaces, they can be used as bioadhesive and surgical sealants. In the area of soft tissue engineering, we have done lots of work on engineering biocompatible and electroconductive hydrogel, which can be used as an injectable form or as an adhesive patch for hard tissue regeneration. Some of these hydrogel were formulated as sprayable biomaterials, which can be loaded with different components, different therapeutic agents, and then we use them for wound healing application. We also work on engineering shear thinning hydrogel, which can be used as bioink to print 3D vascularized tissue. In the area of surgical bioadhesive and sealant that I would like to cover more during my presentation today, we have done lots of work on using elastin-based biomaterials to form bioadhesive and surgical sealant, which can be used for sealing internal organs such as lung and blood vessel. And some of these hydrogel were formed in a transparent and a specific shape that can be used to seal different defects on the cornea. We also form osteoinductive bioadhesive, which can be used for treatment of different dental diseases, such as peri-implantitis. Once we form this hydrogel, we collaborate with clinicians to test their efficacy in small and large animals. And of course, the goal in my lab is to translate some of this technology from the lab to clinic. My lab has a long-term interest in engineering elastin-based biomaterials for tissue engineering and surgical application. We have been able to form this elastic substrate by using different engineering tools. And the benefit of these elastin-based biomaterials is that they can provide elasticity, which is very similar to the native tissue in a body. As you may know, elastin is an insoluble natural protein, and it is the key extracellular matrix protein of our connective tissue. It plays an important role in tissue biomechanics, but one of the problems with elastin is that it's insoluble. So in order to form elastic hydrogel, we should use soluble form of elastin, including animal-derived elastin, different sequences of elastin-like polypeptide, or the monomer of elastic fiber, which is called torpoelastin. Few years ago, in collaboration with Dr. Khadem Hosseini and Dr. Wyss from the Sydney University, and in close collaboration with our clinician collaborator, we formed an elastic sealant and bioadhesive, which can be used to seal different defects on the lung. So this sealant was formed by using torpoelastin molecules. This is recombinant human torpoelastin. And through a one-step reaction with methacrylate anhydride, we form metro or methacrylated torpoelastin, which can mix with a photoinitiator, and by exposing to light for a very, very short period of time, you can form a highly elastic hydrogel. So we show that Metro is elastic and adhesive to the wet surfaces. It has tunable physical properties. And we also show that it promotes tissue regeneration and wound healing over time. It's biocompatible and biodegradable. And you can also cross-link it in less than a minute, and it has ability to immediately stop air and fluid leakages. About the tunability of Metro, for example, here are some data based on burst pressure of Metro when we use it to seal different defects. We show that by changing the polymer concentration or the degree of the functionalization in Metro, you can tune the burst pressure of this biomaterial. We also compare our data with a different type of surgical sealant. Avicel is a fibrin-based sealant, Co-Seal is a PEG-based, and also we use Progel, which is the only FDA-approved sealant for lung application. And we aim to engineer a formulation that has higher burst pressure as compared to commercial product. Once we optimize the properties of Metro in terms of adhesion, mechanical properties, we then test it in vivo. We always start with the small animals. Here is a rat lung incision model where we create a defect on the lung. The polymer was delivered as a solution, and then we cross-link it with light. And then here is the defect that was sealed with Metro. And then following this, we did ex vivo test, and also after 4 days, we measured the burst pressure of the lung by using this device. We apply air inside the lung, and then we measure the burst pressure by using a sensor. And then here we show that based on the day 0 ex vivo data, Metro sealed lung had higher burst pressure compared to conventional techniques that are used for sealing a lung. And also after 4 days, the burst pressure of the Metro seal lung approached healthy lung, confirming that Metro helped regeneration and repair on the defect side. We then, next to our in vivo experiment, these experiments were performed in collaboration with Beth Israel Hospital at Harvard Medical School. And our goal was to see the efficacy, to investigate the efficacy of this sealant in a porcine lung model. We used 2 formulations of the sealant. One was based on elastin and one was based on gelatin. And here is the video show that the defect was created on the lung. And then we check the quality of the defect. And after we observe like bleeding and leakage from the defect side, we apply our hydrogel. So the hydrogel was applied as a precursor solution. And once we apply it on the lung, we cross-link it by applying light. Okay, so this light exposure for less than 1 minute solidified the hydrogel on the surface of the tissue. And then we check the air leakages from the defect side. So by connecting the line to a ventilator and applying liquid or PBS on the surface of the sealed defect to ensure that there is no leakage around the defect site. So here we observe complete stop of blood or air leakages from the sealed defect. So once we confirmed that there is no leakage, the chest was closed and we kept the animals for 2 weeks. And here, this picture, you see the defect that we create on the lung. And then here is the polymer that photopolymerized on the defect side. We did ultrasound every 2 days to ensure that there is no leakage from the defect. And with the histology and immunostaining analysis, we show that the tissue repair over time, and we didn't observe inflammation around the biomaterials. So with this experiment, we confirmed that Metro is effective to seal a smaller size defect on the lung. But in many cases, most of the time after surgical procedure, we should deal with larger defect with extensive bleeding. So to seal these defects for internal organs, in addition to adhesive properties, we need to engineer biomaterial that has also hemostatic characteristics. So to improve the properties of Metro in collaboration with our clinician at a UCLA Medical School, we use our optimized formulation of Metro that provided us with highest adhesion strength to the line. And then we combine it with nanoparticles. These are laponoid nanoparticles that are negatively charged, and they can form electrostatic interaction with Metro. Once we combine them together, we can polymerize them the same way that we did for Metro on the defect side. And then these laponoid nanoparticles can cause blood coagulation on the defect side. Therefore, it can stop bleeding once we apply the biomaterials. Before we move on to in vivo test, we set up an ex vivo test where we use explanted porcine lung, and then we create a larger size defect, apply our biomaterials, connect the lung to the ventilator to measure the burst pressure. And here are some preliminary data showing that as we introduce laponide in our hydrogel, the burst pressure improved, and also the burst pressure for the laponide combined with Metro was higher than co-seal. We next test the efficacy of these biomaterials for blood coagulation. So we start with in vitro experiment. This is a standard in vitro blood clotting assay where we expose different formulation of the biomaterials to fresh human blood. We have our controlled samples that there was no biomaterial in the wells, and then we use Metro with different concentration of laponide, and we also use Surgisil, which is a commercial hemostatic agent. And then once we expose fresh blood to this formulation, we measure the clotting time. We also collected the clot from these wells, and we measure the weight of the clot. And then here we show that as we incorporate laponide in Metro, we have a drop in clotting time, and it approached Surgisil. And we also have an increase in the weight of the clot that we collected. And for the Metro, for example, containing laponide concentration more than 1%, we had a clot mass similar to the Surgisil. So we then next move to our in vivo experiment. For the in vivo experiment, we use a liver bleeding model, and then we use our optimized formulation of a nanocomposite hydrogel, and our control was Surgisil. And for this experiment, we create a defect on the liver with a biopsy punch, and then we seal the defect with our hemostatic biomaterials, and then we use filter paper to collect blood from the sealed tissue. And here is the mass of the blood that we collected, showing that for the group that there was no treatment after 1 minute, and after 10 minutes, we have higher blood mass collected as compared to the Metro laponide and Surgisil. And also for the Metro, as compared to the nanocomposite hydrogel, the nanocomposite was more effective than Metro without nanoparticles. So now that we have our optimized formulation and it is comparable in terms of hemostatic properties to Surgisil, it was important for us to ensure that the addition of this nanoparticle will give us hemostatic properties, but it won't affect the biocompatibility of this biomaterial. So we run this experiment in vitro and in vivo to check the biocompatibility. Our group was Metro combined with optimized concentration of the laponide. We culture human lung fibroblast cells on the surface of this sealant. And then here we confirm that the cells remain viable on these biomaterials. They can spread and also proliferate and has proper metabolic activity as we culture them on this substrate. We also did subcutaneous implantation. Both formulation, Metro with and without laponide, were implanted in rats subcutaneously. And then we show that over time, the hydrogel can degrade, it become more porous, and we observe tissue infiltration and cell infiltration inside the implant. We also didn't observe significant inflammation around the implant for both formulation Metro and Metro with laponide. So our next step here, now that we confirm this material has hemostatic properties and it is biocompatible, now we are working with Professor Cameron at UCLA Medical School to test the optimized formulation of this bioadhesive for sealing internal organs with extensive bleeding. Our next experiment is to test this material in a liver bleeding model in pigs. In addition to hemostatic and adhesive biomaterials, another area that we work in the lab is to form this bioadhesive with immunomodulation characteristics. So to do this, we use a nanoparticle platform which is loaded with miRNA. Once we form these nanoparticles, the base of the nanoparticles is hyaluronic acid. So once we form these hyaluronic acid-based nanoparticles loaded with miRNA, we can combine them with any kind of photocurable bioadhesive. And by mixing them and applying them on the wound, we can form an adhesive hydrogel that can also release this miRNA over time to control inflammation on the wound site. So in this project, we loaded different concentration of miRNA inside the hydrogel, and our goal was to preserve the adhesive properties and mechanical properties of the gel once we incorporate these nanoparticles. Here we show that by adding different concentration of them, the adhesive strength won't change, and also it was higher than Avicel and Cocil that was used as control in our experiment. Then we test different formulation of the hydrogel in our wound closure model in mice. So here we tested different group. These are the wound that we created in the back of the mice, and then we apply PBS as our control. This is a PBS treatment, and then we apply our hydrogel without microRNA and our hydrogel containing optimized concentration of miRNA. And then once we look at the wound closure, we observe that the wound closure percentage was higher for the group treated with miRNA as compared with just the bioadhesive and the PBS group. And if you look at the H&E staining, you see that for the group containing miRNA, we have better healing, better wound healing over time. And also based on immunostaining, we observe higher degree of vascularization as we incorporate this miRNA inside our bioadhesive hydrogel. Therefore, by combination of miRNA nanoparticles in a bioadhesive biomaterial, you can control the immune response toward regeneration and vascularization. So this was one modification that we did in our adhesive biomaterials. But in addition to inflammation, another complication during the wound healing process is infection. So to form these biomaterials antimicrobial, we also work on combining different type of antimicrobial peptides in our hydrogel platform. In this specific project, we used two different extracellular matrix protein in our skin. One is elastin, and the second is gelatin, denatured form of collagen. By combination of these two polymers, we could form an adhesive hydrogel with mechanical properties and elasticity similar to the native skin. And then by incorporation of an antimicrobial peptide, which can be conjugated to the backbone of the polymer, we can make this material antimicrobial. So once we add antimicrobial peptide, they can be sprayed on the wound and cross-link with visible light. And then we show that the antimicrobial peptide can release over time from the biomaterial. And in this way, it makes it antimicrobial against both gram-positive and gram-negative bacteria. So one unique aspect of this platform is its tunability. Basically, by changing the concentration or the ratio of torpoelastin and gelatin, you can change all the physical properties of the hydrogel as expected. By increasing torpoelastin, you can increase the elasticity or extensibility of the hydrogel. By adding more gelatin, you can make the hydrogel stiffer. You can also play around with the degree of the swelling and degradation rate of this biomaterial. Therefore, based on the final application, you can tune these properties and use it for a wide range of application in tissue engineering. Another platform in my lab focused on the use of this bioadhesive material for cardiac or neural tissue engineering application. And one additional characteristics that we need here is, in addition to biocompatibility, adhesion, and capability to regenerate tissue, we also need to make these biomaterials electroconductive. There are many research and there are many papers in literature based on the use of different type of conductive polymer or conductive nanoparticles, which can be combined with a different biopolymer to form electroconductive hydrogel. But there are two limitations with these techniques. One is the level of the conductivity that we achieve. It's usually less than the native tissue conductivity. And the second is as we add more nanoparticles or conductive polymer in our hydrogel, at higher concentration, they are not biocompatible and they can cause cytotoxicity. So to address these problems, we look into a more biocompatible source to form this electroconductive hydrogel, and we use ionic liquids. Ionic liquids are organic salts that are liquid below 100 degrees. They have high water solubility. They have high ionic conductivity. And cooling-based bioionic liquid are highly biocompatible. So in the lab, we use a bioionic liquid, and then through a one-step reaction with acrylic acid, we form acrylated bio-IL. And then this acrylated bio-IL can combine with any type of photocross-linkable biomaterial to form bio-IL functionalized hydrogel with tunable electroconductivity. So by using this chemistry, we've been able to form conductive and adhesive patches that can be used for heart regeneration. You can place a patch on the heart, cross-link it with light, and then it adhere here, and it helps regeneration over time. We can also use injectable hydrogel that can be injected directly to the heart or to any type of tissue and photocross-link to help with the regeneration. So the way that we synthesize these cardio patches, we use a biopolymer. In this example, we use gelatin methacryloid or GelMA. We use electrospinning to form a fibrous patch based on GelMA. And then once we form this patch, we soak them in Ergocure solution. It's a photoinitiator. And then by adding bioionic liquid, and after photocross-linking, we form these electroconductive patches with tunable properties. Here we show that by changing the polymer concentration that we use to form these patches, and also by changing the concentration of bioionic liquid that we added on these patches during photopolymerization, we are able to change the elastic modulus of the hydrogel. In addition to that, we have a very good control on the electroconductivity of the hydrogel. As expected, when we add 100% bioionic liquid, we had higher patches with higher level of conductivity. As we decrease the concentration of the bio-IL, we have a drop in the electroconductivity of the biomaterial. So you're able to change the conductivity of hydrogel in a wider range as compared to the techniques where we use nanoparticles at lower concentration incorporated inside the biomaterials. So once we form these electroconductive patches with different level of conductivity, we culture cardiomyocyte on these hydrogels. These are primary cardiomyocyte extracted from rats. And after we culture the cells on this substrate, we measure the beating frequency over time. And here the blue line is the non-conductive hydrogel. The yellow line is the highly conductive hydrogel, and we have intermediate conductivity by adding 66% and 33% bioionic liquid. We culture the cells on all these substrates, and here I put a video from the non-conductive patch. This is the blue line. We had some beating, but the cells were not beating circorony. However, when we cultured the cells on the conductive patch with 66% bio-IL, we had higher beating frequency and more circoronized beating of these cells as a culture on the substrate. So the cells respond to the level of the conductivity in the substrate. So to better understand this, we did immunostaining for different cardiac marker. So we did a staining for sarcomeric, alpha-actinin, connexin 43. And then on the top, you see the substrate without bioionic liquid. This is the corresponding to the blue line in this graph. And then the second figure is the second panel is the hydrogel seeded with the cells, and the hydrogel containing 66% bioionic liquid. And then as you see here, when we use electroconductive substrate, we had higher expression of connexin 43 as you see in this quantification data. And this increase in the expression of this protein correlated well with the enhanced beating frequency of the cells on this substrate. So the cells respond to the level of conductivity, and at the specific concentration of bioionic liquid, we observe a better function of these cells in vitro. Next, in collaboration with Dr. Ardehali at UCLA Medical School, we try to see the effect of conductivity of the substrate in vivo. We start some preliminary experiment where we use our optimized formulation of the patch based on in vitro cell study. We photopolymerize it on the surface of the heart, and in this video you see the patch after 3 weeks remained on the heart, and then we did histology analysis, and we also measure the function of the heart over time. And based on this experiment, we observed that for the conductive patch, for the group that treated with conductive patch, we had better regeneration and increased heart function as compared to non-conductive patch, and also the control, which is untreated heart. So with this preliminary data, which is encouraging, this was done it was a mice model. So our next step is to form larger patches and test them in using an MI model in pigs. So these are our ongoing experiments that hopefully we get data soon. So as another part of my presentation, I would like to highlight some of our work in engineering drug loading technology for the management of ocular injuries. So I think this is more interesting for this group listening to this presentation, because we started a company based on this technology, GelMedics, which in collaboration with Professor Donna at Harvard Medical School, and the GelMedics platform focus on engineering drug-eluting hydrogel in combination with different nano delivery platform to treat ocular injury and incision. So the focus of GelMedics is to use hydrogel as a matrix for the delivery of the drug into the eye. So ocular defects can be due to physical trauma, burns, inflammation, or infection. And there are different treatment options for these injuries. One option is suturing, which is applicable for injuries with a minimal tissue loss. Another technique is grafting and transplantation for the injuries with significant tissue loss. And in most of the cases in emergency setting, the clinician use cyanoacrylate glue for fast closure of the wound. However, all these techniques suffer from several limitations, and one limitation is vision problems. There is a risk of infection for the grafting and transplantation. There is a need for donor tissue, and also high risk of immune rejection. And in the case of cyanoacrylate, the limitations are low transparency, rough surface, very rough surface. There is no cell integration inside cyanoacrylate, and also there is a risk of infection and significant inflammation. So there is a big problem in treatment. We have inadequate treatments for the cornea injuries. So that is one problem that we have as a clinical problem in eye treatment. The second problem is effective drug delivery to the eye. It's very challenging, and most of the time drugs are washed out without their penetration inside the eye. If you look at this diagram, you see that on the surface of our eye, we have different layers. We have corneal epithelium, and then we have mucus layer, we have an aqueous layer, and we have a lipid layer on the top. And the presence of these layers form a barrier that protects our eye. But at the same time, the presence of this layer will not let the drug penetrate inside the eye. So this has been one of the limitations in effective drug delivery to the eye. Another limitation is drug treatment regimens that are often complicated and expensive. For the treatment of most of ocular diseases, we need to use a multiple drug combination with different daily doses, and most of the time it's very difficult for patients to administer and comply. So to address all these limitations related to the sealing the defect, closing the wound, and also drug delivery, we come up with a platform technology based on photocross-linkable biopolymers that can be optimized to have an exceptional tissue adhesion basically to the cornea tissue. With this, we have been able to optimize the physical properties of this hydrogel in a way that they mimic the native corneal tissue. And also for the drug delivery part, once we form this bioadhesive material, we also form different nano-delivery platform which can be used to control release of the drug over time. And once we form these nanoparticles, they can be combined with the bioadhesive pre-polymer solution. And then the solution can be applied on the defect side on the cornea by using an applicator. We can use a contact lens as an applicator. Once we place the contact lens on the surface of the eye, we photopolymerize the hydrogel, and after that, the contact lens can be removed, and then we will have an adhesive layer of the hydrogel containing drug. So this is very easy, and the application is fast, and we can have control on the dosage and on the delivery of the hydrogel. And the packing is like you're just having the contact lens, the pre-polymer solution, a small light source for photo cross-linking. And another technology in gel medics focusing on using this hydrogel, but this time we use it as a preformed pellet. So basically the pellet can be loaded with our drug, and then we can insert it into the eye, and the drug can release over time from the hydrogel in a controlled manner. So with this technology, we can address significant problem in ophthalmology, including inadequate treatment of cornea injury and complicated and expensive drug treatments that are used in clinic now. So here are some initial data that we started in our academic lab based on the use of a gelatin-based bioadhesive. We call it gel core, gel for cornea repair. We optimized the formulation of the gel core for rapid repair of corneal stromal defect. Basically, gel core can be injected inside the corneal defect and photopolymerized. Once the hydrogel is solidified, epithelial cells can grow on the surface of the hydrogel, and the hydrogel can degrade by time, and we observe tissue regeneration over time. So in this paper we published a few years ago, we show that the properties of gel core can be tuned by changing the photo cross-linking time or the composition of the hydrogel. And we can reach adhesion strength higher than commercial bioadhesives. And we also apply an optimized formulation of gel core in vivo. This time we use we create an incision on the rabbit cornea by using a biopsy punch. Once we form the defect, the hydrogel was delivered as a liquid, and then we photopolymerize it by visible light. And here is the defect that we seal it with our bioadhesive material. And then we observe progressive wound healing over time once we apply these biomaterials on the surface of the cornea. So here is the video from our in vivo test showing that the adhesive material is cross-linking on the cornea, and then we show the firm adhesion to the cornea tissue. So the hydrogel can remain on the cornea and help with the regeneration and repair over time. So with this paper, we show that gel core has tunable properties. It can be used to seal stromal defect on the cornea. However, when we are dealing with larger size defect like laceration, and also when the eye is inflamed or infected, gel core is not effective. So in the next step, we designed a new formulation of the bioadhesive with improved retention for ocular drug delivery. So our new formulation is based on an adhesive biomaterial. We call it gel patch, and gel patch is made of different polymer that combine together at a specific concentration and ratio to form highly adhesive biomaterial with proper viscosity for the injection. And then once we form a gel patch, in parallel, we engineer particle-based drug delivery platforms focusing on micelles for delivery for the delivering of anti-inflammatory drug, and nanogel for the delivery of the antibiotics. Once we form these nanoparticles and we optimized gel patch formulation, in our third step, we incorporate our drug-loaded nanoparticles into the gel patch. And then here are some targeted or optimal parameters for the gel patch formulation. We use experimental design software, JUMP, to optimize different properties of the gel patch. And At the beginning, we summarize what are the required properties for our gel patch formulation. Basically, there should be some characteristics at the liquid phase, and some specific characteristics are required in the solid phase. Basically, for the pre-polymer solution, our goal was to engineer a formulation that can easily apply, and it has a proper viscosity that we can inject it. So therefore, we look into how we can optimize the composition of the hydrogel and how should be the rheological properties of this material. And once the hydrogel solidify after photopolymerization, it's important to have adhesion strength, enough adhesion strength so it can retain on the ocular surfaces. We want a hydrogel formulation that swell less than 20% to prevent shape deformation. And also the Young modulus and extensibility of the hydrogel is extremely important, because the hydrogel should be flexible enough to for the shape accommodation and curvature formation, and at the same time it should be stable at least for a month in vivo to have a drug release profile over 1 month. So by knowing these characteristics, we design our gel patch that give us all these properties. And then once we form gel patch with optimized formulation, we also work on engineering micelles loaded with anti-inflammatory drugs. So here we use loteprednol, LE, that this is hydrophobic drug that incorporated inside these micelles. And once we form these micelles, the micelles was combined with our hydrogel formulation and photocrosslink to form these drug-eluting biomaterials. We look into the release profile of the drug from these hydrogels. On the top, you see gel patch combined with micelles loaded with LE, and then we observe complete release of drug 100% over 2 weeks. This is the profile that we want for the treatment of inflammation in the eye. But if we put free drug inside gel patch, we also have control release of the drug, but we cannot achieve 100% release over time. We also look into the different physical properties of gel patch. Our goal was to see not significant changes in the adhesion and mechanical properties of the gel patch once we introduce our nanoparticles. And we also did in vivo test using subcutaneous implantation model to see the degradation rate of these gel patch formulation containing drug over time, and also look at the inflammation and in vivo biocompatibility of this formulation. So now we are testing this hydrogel in animals with inflamed eye to see their efficacy in treatment of eye inflammation. We also did some work on making this hydrogel in a way that they can be used for treatment of the infected eye. And this time, we form micelles loaded with antibiotics. Once we create these micelles, they can be incorporated in the hydrogel to form these antimicrobial biomaterials. And then we show that as we incorporate these micelles loaded with antibiotics inside the gel, it's still effective to seal incision with different sizes on the cornea. So as you see here, for hydrogel with and without micelles, we have the same burst pressure for different incision sizes. So therefore, this platform can be used as a matrix for delivery of different drugs into the eye. And it can be used as a photocross-linkable hydrogel that can adhere on the surface of the eye, or it can be used as a pellet, which can be injected in the eye for controlled release of drug over time. So as a last part of my presentation, I hope I'm not over Homan or Mohsen. Can I present this, or you want me to finish up here?
Yes, please go ahead. We are enjoying. Please.
Okay, great. Thank you. So the last part of my presentation is based on another biomaterials that we designed with my great colleagues, Professor Intini. We started this project when I was in Boston, and he was at dental school at Harvard. And we designed a platform technology for the treatment of dental diseases. So as you know, dental implant constitute the standard of care for tooth replacement. However, by placing 12 million implants per year, post-operative condition that affect peri-implant tissue has become a big concern. And one of the major problems is peri-implantitis. That is a disease that starts with the infection around the implant. And if we cannot control infection, it can also cause bone loss over time. So therefore, when you design a biomaterial for the treatment of the peri-implantitis, your biomaterial should have dual function, antimicrobial and bone regenerative capacity. So the treatment option for the treatment of the PI or peri-implantitis currently is mainly based on guided bone regeneration. Different type of, for example, bone graft are used, or biomaterials that can deliver also inductive growth factors such as BMP2. And one of the most popular one is Infuse that they use it in clinic. So when we look at different treatment options, including Infuse, there are some limitations. So basically, Infuse can help bone regeneration, but it doesn't have any antimicrobial properties. Therefore, they have to use it in combination with different type of antibiotics. Okay, so the treatment that has like an antimicrobial effect for the implant for peri-implantitis treatment, they don't lead to bone regeneration. So our goal was to design a strategy or a biomaterials that has three properties together: adhesion, antimicrobial activity, and also inductivity. So with Professor Intini, we designed our biomaterials when I was at Northeastern, and then once I moved to UCLA in collaboration with Dr. Piri at UCLA School of Dentistry, we were able to implant our optimized formulation in mini pigs and show its efficacy in large animals. So the bioadhesive design is basically the first part of it is the bioadhesive material, which is biocompatible. It can degrade into non-toxic bioproducts, and then we can deliver it as a liquid precursor solution around the implant, and then we can photocross-link it by using a dental curing light. And then once it's photocross-linked, it can have strong adhesion to both implant surface and also to the soft and hard oral tissue. So once we form our bioadhesive formulation to make it antimicrobial, we combine it with our antimicrobial peptides. It can be used as an effective antibiotic-free approach. We have shown that in our publication, antimicrobial peptides are very effective at low concentration. They are biocompatible and they won't cause antibiotic resistance. And then to make this biomaterial also inductive, we combine them with also inductive nanoparticles, laponide, and these are biocompatible nanoparticles that can degrade into non-toxic bioproducts, and we can use it as a growth factor-free approach to make our biomaterials also inductive. So we optimize the concentration of all these elements to form this photocurable hydrogel, which can be injected around the implant. And once we inject it as a liquid, it can fill all the voids and cavities here. And then by using a dental curing light, you can photopolymerize it. So here are some data based on ex vivo test. We wanted to measure the adhesive properties of this hydrogel to the gingiva tissue and also to the titanium. Here are different formulation of the hydrogel. This is the gel without laponide and without AMP, and then we added different concentration of laponide and antimicrobial peptide, and we measure the adhesion strength of the hydrogel on the gingiva tissue based on wound closure test, and on the titanium surface based on lab shear test. And then we show that the adhesion increase as we introduce these nanoparticles inside our hydrogel formulation. We also did in vitro antimicrobial test on these bioadhesive against P. gingivalis, and then we show that as we introduce antimicrobial peptide in our hydrogel, we can form an antimicrobial adhesive biomaterials. Here is a representative SEM image from the bioadhesive without AMP, showing that the bacteria can grow inside the gel. And then once we introduce AMP at optimized concentration, we can prevent bacteria infiltration inside the bioadhesive. We next test the in vitro cytocompatibility of the bioadhesive. In this experiment, we culture human MSCs on these hydrogels. The control was a gel without nanoparticles with AMP, and then we introduced different concentration of bioadhesive formulation with different concentration of nanoparticles. And then we show that for all formulation, even the highest concentration of nanoparticles, we had high cell viability both on day 1 and also day 5. The cells could grow on the surface of this hydrogel and proliferate over time. We also look into the differentiation of MSCs once we culture them on these hydrogels. We look into the expression of different bone-forming markers, and then we show that for different group control, different concentration of nanoparticles, and also our control. Another control was BMP2 added to the culture media. And then with our experiment, we show that higher concentration of nanoparticles resulted in higher expression of these genes, and it was very close to the control that was the addition of the BMP2 in the media. So our optimized formulation was the hydrogel that has higher concentration of nanoparticles. We next perform an in vivo experiment using Calvarian defect model. This was done in Professor Intini lab, where we created defect, Calvarian defect on mice, and then we applied 2 different formulations of the hydrogel. One was formed by using 15% gel, and the second one was formed by using 7% gel. So I call them a stiffer and softer matrix. For 7%, the modulus was lower as compared to 15. And then our goal was what happened to the bone regeneration when we implant different formulation of the hydrogel. And here on right side, this is the defect we create. This is untreated after 42 days. And these are the defects that the left one treated with 7% hydrogel and the right one treated with 15% hydrogel. So after 42 days, we have higher bone formation for the group treated with stiffer hydrogel. So to quantify this data, we did a Microcity analysis. This is the defect we create at day 0, untreated group, day 28, day 42, and the group treated with softer hydrogel versus a stronger hydrogel. And then based on the quantification data for bone surface area and bone volume, we show that as we use hydrogel with higher stiffness, we had higher bone formation compared to the softer hydrogel, and both of them were better than the control group. So with this experiment, we confirmed that using hydrogel with 15% polymer and higher concentration of nanoparticles is more effective for bone regeneration. So in our in vivo evaluation of this bioadhesive, our experiment was based on a model of induced peri-implantitis model in mini pigs, and this is a very long study. It took us a year to complete. It is started by tooth extraction in mini pigs, and then we waited for 3 months for the healing process. And after 3 months, the implant was placed, and then we had 2 months of healing process after placement of the implant. Then we induce peri-implantitis by putting a ligature around the implant. And then after 10 weeks, when we observed the formation of the peri-implantitis disease, we started our treatment. Our treatment started by applying our hydrogel around the implant, and we also use a commercial bone graft to compare with. And then once we treat the pigs, we waited around 4 weeks, and then during this time, we did different evaluation, H&E staining, microcity analysis, immunostaining. We also get sampling for the microbiome study. And then here are some results that we obtained. This is the implant that we treated with bioadhesive group. This is the hydrogel around the implant. It remained around the implant and helped bone regeneration. And this is our group that treated with the bone graft. And then when we look at the bone formation around the implant, the hydrogel group was more effective as compared to the bone graft and the control group, which was untreated group. So we have lots of data with a microcity analysis and for the immunostaining that hopefully it will be published soon. So these experiments were performed at UCLA Dental School with Professor Piri teams. So to finish up, I would like to acknowledge my great students and research team and all the collaborators who help us significantly in different projects. Our funding sources, NIH for the sealant project, BOD for the Ocular project, and also American Heart Association for the electroconductive patches. And thank you very much for your attention, and I'm happy to answer your question.
Thank you so much, Professor Annabi. This is a great, fantastic presentation with lots of application. Yeah, we really enjoyed, and we didn't want to really, we wanted to continue this presentation. So I just want to ask you before going to the question, how much time do you have to stay with us to answer the questions?
I have time till 10:30, so we have around 30 minutes. If it is okay.
So because we have several questions in the Q&A box, and I saw Mohsen also was writing many questions on the other side, and I have also my own. So we stay a bit. So let's start with the audience question.
Sure.
Amir is asking.
So can I ask a question?
Yeah.
Should I close my share screen, or what do you want me? Because I don't see anyone. I don't see
you
even
can.
So
yeah. It's better to stop it. Stop the share screen. So
let me see how I
in the middle, I think. Yeah, okay. There. Yes, exactly. Okay, that's great. So now we feel better. So we see each other.
Exactly.
Yeah. So Amir is asking that. You talk about many adhesive, bioadhesive material. First of all, he's thanking you for the nice presentation, which was very illustrative. And he's asking, he has many questions. I'll try to combine. How long?
Who is Amir? Can you also tell me?
Amir Jafari, not, yeah.
Okay, good.
How long or how many days it takes for this material, the elastin-based bioadhesive, to do to do regeneration or tissue regeneration. And after regeneration, is there any scar tissue or any sign of scar tissue in the on site? And I think maybe we can answer these two, and then we move forward with other questions.
Okay, thank you, Amir, for asking this great question. So about the regeneration for the elastin-based biomaterials, elastin has a slow degradation rate as compared to other biopolymer, right? It stays longer, but it also can degrade by enzyme as we implant it. So we can tune the degradation rate. It depends on how much polymer you use or how much cross-linking you do when you make this bioadhesive. So for our formulation, it become porous, but it still stay on the defect after like 3 months or based on subcutaneous implantation. So about the scar tissue, we didn't study that because it was for internal application on the lung. It was biocompatible, so it was a thin layer on the lung. So even if it stay longer, we don't mind, right? So as long as it eventually degrade, we didn't do anything to control the degradation. But the scar tissue formation, we didn't look into that. We checked the inflammation. There was not much inflammation around the implant, both in pig model and in the rat model. But we didn't do extensive study to evaluate the scar formation of elastin.
Thank you so much. Nureddin is asking, in immunomodulatory hydrogel, what is the optimal dose of mRNA to get best result in terms of anti-inflammatory effect without inhibiting regeneration?
That's a great question, Nureddin. I don't have on the top of my head how much we add, but in the paper that we published in small, we show the effective dosage of that. So sorry, I don't remember exactly the number, but we look into different concentration of miRNA, and then we look at the release, what will be the release profile over time. And then we optimize the concentration that gives us highest wound closure percentage and also the highest degree of the vascularization. Before we do those studies, we did lots of in vitro tests with immune cells in vitro, and then we with the Transwell, we expose different concentration of the nanoparticles to the cells to see that how they switch, the macrophages switch from M1 to M2. And then we analyze that with PCR. Once we confirm the dosage that is effective for this polarization, we move on for our in vivo test based on that specific dosage. So if you email me, I can get back to you and I can send you the paper.
Thank you so much. I think he can also come by and get the hard copy from, because he's in the same building probably. In electroconductive, Nuruddin asking again about the other project that was about cardiac patch. In electroconductive patches in vivo experiment, the size of the patch relative to the mouse heart may be big if you think of clinical application limited to the infarcted area. Did you observe any arrhythmias?
We didn't observe arrhythmia, but it was very difficult to do that test, to be honest, because the heart is very small and it was even difficult for us to place the patch on the heart. So we didn't observe arrhythmia because the patch that we use has somehow similar electroconductivity to the native heart. I can imagine if we use the one with highest degree of bioionic liquid, we would observe arrhythmia. We didn't test that, but we optimized the formulation that has similar conductivity. We didn't observe arrhythmia, but I can tell that doing that experiment was not easy. That's why we have to move on to larger, animal model. So pig is or sheep is the best, species to test that, and that is what we want to do. And we are waiting for NIH to fund our project so we can do it with Professor Ardehali. But yeah, definitely.
That's great.
Is a great question. But the good things is with this, you can make patches in any size and any shape that you want. You can make them larger. My students used to make larger patch and then we cut them in small pieces for the rat experiment. But I imagine for pig, we may need to use larger patch, but we have ability to make different sizes and different shapes as needed.
Yeah, I can imagine that the use of ionic liquid, help also solving the problem with nanoparticles distribution in the other downstream, tissues, because there are lots of debates, if these nanoparticles go and accumulate in other upstream or downstream tissues. But
exactly, and another problem as you work on this system, I know one of the problems that we have with nanoparticles, we don't get good conductivity. We have to use large amount of them in biomaterials, and then most of the time, at least in our system, we get problem with aggregation in polymers and lots of complication. And at the end, you can't tune even the conductivity because it is a very limited range. But with this, you can finely tune the properties. You can make many very conductive biomaterial that can be used for most and like an application with this excellent work on flexible electronics that he does, or for regeneration by controlling it at lower level, similar to the native tissue. I think it's a strong platform, and now we use it for even nerve tissue regeneration application. We collaborate with Stephanie in our in bioengineering at UCLA to see that how we can use it for the neural tissue regeneration also.
That's great. Thank you so much. Mohsen, could you please, when I ask the question, you can just remove them from the list so I can organize this question. So we go to Ali question. He's asking about the stability of these hydrogels and how to increase its stability, in vivo or probably in clinical application or off the shelf. I don't know exactly what he meant, but
so stability are two things, right? So I'm learning this now for the company. So one is stability of the material before you deliver it, before you use it for your application. You want to see that for how long it is stable, right? So before use. So for that, we have to do lots of tests, and then this is what we want to do. We have to see that what is the effect of temperature, or what is the effect of light and other things that may affect the stability of the biomaterial. And these are a year or 2 years experiment, then you explain what is the shelf life of the biomaterial that you want to make. So that is one side. And then stability has another meaning. I don't know which one Ali is referring. So when you implant it in vivo in the body, how long it stay until you see the function that you want. You want to seal a defect, so it should stay for a couple of days. Or you want to deliver anti-inflammatory drug in the eye, so it should stay for at least 2 weeks. So if you mean stability by that also affect the efficacy of your biomaterial, it depends on your chemistry and the biomaterial design. So, for example, one simple way to do that is, let's say you have a hydrogel that degrades fast, but you want to use it for longer time in vivo. So you can use polymers that can slow down the degradation. And in this example, for example, PEG can add to your polymer to extend the degradation rate in vivo. So by the composition of the biopolymer and also by the way that you functionalize it, higher degree of functionalization versus lower, so you can tune the stability in vivo. So the best way is to see what you want and then design your biomaterial in the lab to target that time frame.
Thank you so much for your great answer. Fatima has a couple of questions. I'll try to combine them together. In general, she wants to know about the the mechanism to accelerate the healing process, how we can accelerate this process, and specifically, she's asking about your bioadhesive compared to the sprayable hydrogel. Mm-hmm. What are the advantages of those that you made or developed compared to the other commercial one? And also, yeah, that's it. So that's.
I have a very short memory. I can answer 2 questions at a time.
Oh, okay. So I'll ask one.
But now I do remember 2 questions from Fatima, right? So these are great questions. So regarding the regeneration, how we target different wound healing process. Basically, wound healing has different stages, right? So it starts with inflammation, and then you have to go through the different stages of the wound healing, and then the first step is inflammation, right? You have inflammation on the wound site. So to control inflammation, you can find strategies to switch macrophages polarization toward healing. So this is the immunomodulatory biomaterial that I introduced. So if you can make inflammatory biomaterial that can control the release of inflammatory cytokines or molecules during the first few days of wound healing process, so you can control that stage. And another stage of wound healing is hemostasis, and then we have also regeneration. So you can see that how you can target different stages of the wound healing, and then by incorporation of different components in your biomaterial that can control by releasing in that specific time frame. Okay, so another thing for wound is infection, right? We always have infection. So if you can find a strategy to incorporate antimicrobial components, zinc oxide, polymer like chitosan, they have antimicrobial properties. We use AMP. That's another benefit. So one thing that you ask, which is a great question about what is the advantage or benefit of our adhesive biomaterial, is that they have multiple function, right? So they can do several things by application of one single biomaterial and a single dose of that, right? So for example, let's say you make a platform that adhere to the wound. It helps regeneration because you use a naturally derived biopolymer, so it helps with the natural regeneration of the tissue. At the same time, you decorate it with different components that can control inflammation. You can make it antimicrobial, and then once you incorporate all these components in a single platform, it helps you to further promote different stages of wound healing. And the goal is to make it faster, right? Because the wounds repair by its own, right? So if you can make this process faster and more effective, I think that is where we look into design of these biomaterials.
Thank you so much. Hafez has a question that also is my question too. So he's also thanking you, everybody thanking you about your great talk. And I just don't repeat that. But Hafez is asking, what is the main adhesion mechanism of the gelatin-based adhesive or elastin-based adhesive hydrogels? Have you done any chemical modification to increase the adhesion of these bioadhesive?
Mm-hmm. That is a great question. There are different bonding that can happen with the tissue. First of all, let me tell you that it's physical and chemical. What is physical? Physical means that when you deliver a solution as a liquid on the surface of the tissue, the liquid can penetrate inside the fibers on the surface of the tissue. As it penetrates, once you cross-link it with light, you have interlocking between your polymer and the tissue. We call it mechanical interlocking, okay? This is physical. In addition to that, we have chemical interaction with the tissue. Sometimes you have the, charge, polymer, for example, bioionic liquid is charged, and then they can make electrostatic interaction with the amino acids on the surface of the tissue. So electrostatic interaction is another one. You can have hydrogen bonding on the surface when you apply these materials with the tissue. In addition to that, there are many ways that you can further improve the adhesion. One way that you can do it, and we have a paper on the review, 2 papers based on dopamine. Dopamine is we can conjugate dopamine on the polymer, and this dopamine can help with the adhesive properties of the biomaterial to the tissue. There are different ways, and I can tell that there are great review articles. I'm happy to send it to you. I forgot the name of the person who asked this question.
Hafiz is asking.
Please send me an email and I can forward you some review papers just looking into the mechanism of the adhesion to the wet surfaces. And there are many things from this MIT group that they form this double-sided tape, and the tape can absorb water on the surface of the tissue, and then it is moisture sensitive, and then it adheres once it absorbs water, right? So there are different mechanisms that you can design to have this high adhesion to the tissue.
Thank you so much. Norbert is asking more about commercialization. Are you planning on commercialization of more products? And how easy it is to start a company in California?
It's not easy. So I have started. So it is impossible almost to do several at the time. So at the same time. So we started up a company with my collaborator, Professor Donna, and it's Gel Medics, and it's based on the Ocular project that I explained. And I can tell you that it's lots of work and lots of involvement. So we are co-founder of the company. We have CEO and CSO and a team who are working on the development. It is out of our lab now. But before the technology licensed to the company through UCLA and Harvard, there were lots of work for us to do it in the lab and to make sure that everything is working and optimized. And then now we transfer the technology to the lab of the company, and that is a lengthy process. So I can tell you that it's lots of work. It's not just the science part. There are many different components of that to see that who are the competitor, how the technology works, how it's better than other things, and your business plan. And it's not a work of one person, first of all. There should be a great team of people who can work closely together on daily basis. They communicate. And in addition to that, it takes lots of time. So for me, I think that is my first experience. So I start with Ocular, but I can tell that I am very, very interested. It's a difficult, but it's a great journey for all of us.
So that brings another question. How do you manage the work and life balance?
It's not easy because for us as full-time faculty, we cannot use our faculty time to do this activity. We have a specific time. For me, it's 3 hours per week or 4 hours per week. But of course, all my weekends. And after 6 o'clock, I will start working on my deck presentation, talking with people. And the good things for me, the company is based in Boston, and I am in California. When I have to do presentation, it's 6 o'clock in the morning, which is 9 in Boston. So I can do my presentation early in the morning, and I start my academic work around 8, 9. But yeah, it is a difficult, very difficult, but I think it's very, very interesting journey. So at least for me, I'm very happy going through that. It's not important what will be the outcome, right? Of course, we want to be successful and the company succeed, but I think through this path, you will learn many different things that you cannot get those experience just by being in academia, I think. So I highly suggest it if there is a chance for you to do that, definitely.
Sure. And we hope to see more companies and more products from your lab. And I mean, because now you're so productive, and this gel medics, probably you will have more products in portfolio later on.
I hope
So,
so.
yeah.
Important things, Homan, is all the technology that I presented for you today. There are patents already, like full patents based on all of them, right? So when you have good data, or when you have an idea that you think it has potential for future use, so it's important to have your patent application before you publish them. So this is what we do always in the lab, that we get the IP, like a disclosure form, and then you have a year to convert it to the full patent. And once you do that, you're safe to publish it or to talk about your technology in conferences. And I think even though if you don't have time to do it at that stage, you can always file your patent application in your institution if you are in academic setup, and you can always keep your eyes open to opportunities for commercialization and see that who might be interested. And of course, team. So as soon as you find the right people to work with, I think that is a good time to get started.
Yes, thank you so much. And Isabel has a question about biodegradation of the adhesive hydrogels.
Mm-hmm.
Have you checked the in vitro biodegradation of these hydrogels?
Elizabeth, that's a great question. So yes, we look into the degradation rate of the hydrogel, both in vitro and in vivo. And what I can tell, we have published papers based on, we put, for example, the hydrogen in enzyme solution. We look at the degradation over time, but you cannot always predict what happened in vivo with those in vitro study, because we never know how much enzyme to add and how to manage the in vitro experiment in a way that it mimic what really happened in the body, right? So in vitro biodegradation data are great to give you a timeline to compare materials, one material versus another. But if you want to get more information on the biodegradation rate of the biomaterials, I think in vivo test is the best way of doing it. And another complication is in vivo, it depends where you implant it, right? Degradation rate in the eye may be different from subcutaneous implantation, right? So you have to implant the hydrogel or biomaterials where you want to use it and see that how it degrades over time. So that is the optimal way, but of course in vitro would help. Depend on the backbone of the polymer. You can run different study. If your polymer is made of proteins that can degrade by enzyme, of course you have to use an enzyme solution that to evaluate that degradation rate.
Thank you so much. Shahrzad is asking about the bioadhesive for bone, for teeth.
Mm-hmm.
So she's wondering if you compare the formation of the junctional epithelium, pre-implant epithelium, in your sample group compared to controls, specifically the needed time for the gingival epithelial cells to migrate to the implant soft tissue interface.
That's a great question, and I am not sure if we did that, because that part, the in vivo, was performed by Dr. Piri, and then they mainly look into the bone formation inside the implant and versus it, compare it versus commercial bone graft. I'm not sure if they specifically look into that. So, but that is a very good point, and thank you for raising that. So definitely I'll discuss that with my collaborator.
Thank you so much. I'm going down the list. I see that Nuruddin said that amazing and inspiring work. Thanks, Nasim.
Thank
and Mustafa is asking, about the later slides that you presented regarding the treated implants. How much cost this type of treatments have compared to the current commercially available dental implants? I don't know if you treated. Yeah, you treated. Yeah.
We treated. We didn't design the implant. Is implant. So we didn't touch the implant. So we inject the gel around the implant. If your question is how much is your gel versus the commercial bone graft, the answer is it's much cheaper, because our base material is very cheap hydrogel, and then the nano clay are very cheap. So the composition of the hydrogel is like, it has lower costs compared to infuse, right? If you guys work with BMP2 in the lab and you know that it's very expensive to obtain those. So this biomaterial can produce at very low cost. So we haven't, we patented the technology, but we haven't looked into marketing or like, business plan for this. But I assume that it should not be expensive based on the source of the material that we use. And of course, the way that you cross-link it, it is the dental curing light. You can find it in any dental clinic, so you don't need even an additional tools for the delivery of the biomaterial.
Great, great, thank you. And Hussein is asking, I think this is the question of Mohsen too, is asking about a sterilization strategy for the adhesive material, and specifically for cardiac patch he mentioned here. Did you prepare the patch in aseptic or GMP condition, and how do you sterilize these bioadhesive?
That's a great question. We didn't do GMP. So of course, in my lab, we didn't have facility to do that. So definitely for commercialization, there should be a specific way for sterilization, like gamma radiation, or like, some approach that wouldn't affect the bioactivity of the scaffold. But for the lab experiment that we did for the mini pig, for the mini pig experiment, or for all the in vivo experiment, we try to make the samples as much as we can in sterilized environments, for example, under biosafety cabinets. And we always filter the pre-polymer solution before we use it for photo cross-linking, and we set up everything under the biosafety cabinet. So some of these material, for example, when you make the patch, you can also sterilize that by putting it under UV, we put it under UV light for 10 minutes, 15 minutes to make sure that it is sterilized. But we don't have any protocols, right, that really help to sterilize it in a level that can be used in human. We don't have problem with contamination, but of course for human use, there should be a specific strategy based on FDA or GMP approved facility that we don't have it in my lab at this stage.
Thank you so much. We have another question from Gajanan. Gajanan is asking about the behavior of the cardiomyocyte on the electrospun fibers or electrospun cardiac patch, I think you showed. Mm-hmm. Can you comment on their behavior or beating properties on these materials? I think you mentioned that, but sure. maybe in brief.
Mm. Yeah. Okay. So when we put cardiomyocyte on this substrate, we saw that the cells responds to the level of the conductivity in the substrate. So for the hydrogel that was medium conductivity, we published that paper. If you email me, I can also forward that to you. So we show that for the patches with medium level of conductivity, the beating frequency and circony was better as compared to non-conductive. In the video that I show you, the left one was the patch without conductivity. The cells beat in different area, because when you isolate cardiomyocyte at the beginning, they are silent, so they don't beat well. So by day 3, they start beating, and then if you put them in a right environment, you see an improve in the beating frequency of the cells, right? So when you expose them to the non-conductive biomaterial, some of them are silent, some of them are beat, and you have this in silico coronary on your patch. But once you introduce them to the electroconductive patches, these patches can help or promote cell-cell communication. So you have higher beating frequency and better circuity in beating as they expose to the electroconductive patches. This is what I show in that page and with the video. And of course, the level of the conductivity shouldn't be also very high, right? So you should have it optimized.
Great. Before going to Mohsen's question, because I know that he has many questions, but thank you, Mohsen, for your patience. Ali Reza is asking, I think you already answered Ali Reza, but maybe we can touch his question about using carbon-based or graphene-based nanomaterials in cardiac tissue regeneration. What is your idea about using this kind of materials as a conductive element?
I have a published paper on graphene oxide and reduced graphene oxide. I think it published when I was in Ali's lab, 2013. And I remember we went through pain with my great collaborator, Suryan Shin. So one of the main problem was when we tried to combine these nanoparticles with the polymer. So our goal was to make electroconductive hydrogel. Aggregation was a main problem. As soon as we introduced them into the polymer phase, if you don't know how to mix them, what condition to use for the homogeneous distribution of these particles, instead of increasing the properties, you will have drop in both electroconductivity and mechanics. And I think one limitation with carbon nanotube or graphene oxide is the dispersion if your goal is to combine it with another polymer. The second problem is limited conductivity. Even if you manage to disperse them very well, you get limited conductivity. So, and you cannot really tune it, right? It is a very, very narrow range. And the third problem is biocompatibility, right? Up to a specific concentration, they are great. But above that, you will have cytotoxicity. So it is not just carbon nanotube and graphene oxide. We also work on, I have a PhD student who finished up his thesis by using this P.PSS. He compared different material. And then for P.PSS, we obtain conductivity up to 0.1%. But as we increase it to 0.3%, the viability drop, right? So you always have a threshold. So these are some limitations. Of course, there are lots of benefits in using carbon nanotube, but because of the biocompatibility and the problem that I just mentioned, I do not like to use them for tissue engineering application, right? So they can be used for many other applications, but in tissue engineering, you should think about all of these parameters when you design your biomaterial.
Thank you so much, Nasim. And Mohsen, you have 2 minutes to ask all your questions.
No worries.
Please go ahead.
Well, first of all, thank you very much, Nasim, for your amazing presentation. You always provide new data whenever I listen to your talk. Very productive. It's fantastic. And as I mentioned, Nasim was my mentor when I was in.
No, that is not true. We were colleagues both. We had a great time at Harvard together. So I learned so many things from Mohsen, and we were great collaborators.
Yeah. Well, thank you, Nasim Jan. And then, I'll try to be as quick as possible. I'll just ask one question about because I have many, but I think one is the most important one at the moment. I noticed that you're using so many different animal models in different applications. Ranging from small animals to large animals, ships in the future maybe, and then on what you're using pigs. So when you start designing your experimental plan, how do you decide what animal is the best for your application?
That's a great question, Mohsen. I don't decide. I collaborate with relevant people who decide about that. So, but of course, this is a great question. So for some of the small experiments, a small animal experiment like subcutaneous implantation or liver bleeding model using rats, all of my students are trained and they can do those tests in the lab. But for a specific indication, when I want to test them in more relevant model, I do collaborate with clinician, and then I learn from them that what is the most common model to be used. For example, for our peri-implantitis project. So when I talked with Dr. Piri at UCLA, so my first question was that what is the most common species to use, and then he said that dogs are great. So and the second is like pigs or mini pigs, right? So there are papers and literature that we can see what is the most common one, and then I think by asking people who are working in the area of the application of your biomaterial, they can give you suggestion of what is the best model for testing of your biomaterial. And some models are not easy to be done in rats or small animals, and it's very challenging. And if even you can do that, you won't get reliable data, right? So it has to be done eventually in large animal model, and the model should be designed by the end user, which are surgeons or clinicians that you are working with.
Okay, that's fantastic. One follow-up question then. So in terms of, so now that you're entering into the translational part of these technologies, would you consider FDA guidelines as well in designing the animal studies? So that's another question that I wanted to know more about.
Yeah, that's a good question, Mohsen. Yes, what we do for the company, we we did tech transfer from the lab. So the company now is working on using, first of all, GMP approved sources for all the materials that we use. So the hydrogel that I designed, it has 3 different polymers, and then they found other source facility that they can provide us with the GMP approved biomaterial to start with. And then for all the experiment that now they are doing, what they do, they use GMP approved facilities in Boston area for both in vitro and in vivo testing. And then what they do, they want to basically replicate what we've done in our research lab, but in GMP approved facilities. So in this way, FDA would accept the data that will be generated. I'm not involved in those parts, but I know that they are looking into where to do those experiments.
Well, we're running out of time.
Our lab, we don't do it in our lab at the company event, so we can't set up those, and it's expensive. So, but you can pay, and then they do the test for you.
Makes sense. No, that's completely. It makes sense. It makes complete sense. So again, I have more questions, but maybe we can talk offline. I know you're busy, and then you mentioned that you have another meeting at 10:30. Thank you very much, Nasim, for accepting our invitation.
Thank you, and please feel free to call me, Mohsen, with your question, always.
I will.
Pleasure to talk with you, and thank you so much. Both, I really enjoyed this presentation, although I can't see people, but I'm happy that they ask lots of questions, means that they listen to my presentation. So.
Thank you so much. Thank you so much. And I just want to say to audience that see you next Wednesday at noon Eastern time, and then we continue this e-series. And thank you, Nasim, for your great talk. And
thank you so much. Thank you, everybody.
I'm a fan of your research, and I have been always following your research, and learned a lot. Thank you, and have a great day.
From all of you. And then one more thing, I have opening for PhD students and
this is great.
Both. I got recently I got a grant from DoD, and we are hiring. So please, if you are interested in this type of translational research relevant to these e-seminars, send me an email with your CV. Okay, and for Mohsen and Homan, I would love to hire your students. If you are graduating any PhD students, just send it to my
Sure. Thank you so much. Of course.
Thank you very much.
Thank you. Take care, everybody, and bye-bye.
Have a wonderful day.
Bye.
You too. Bye-bye.